My name, thanks. My name is Carolyn Risley, and I am your host today. If you have not already, do not forget to silence your phones. Just as a disclaimer, this event is being recorded. This is so we can post all of the videos on YouTube later for anyone who missed the seminar. We're a couple minutes behind schedule, but first we're going to have the announcements, which I'm doing right now. Then we'll be followed by an introductory trainee talk, the featured talk, and then, as always, free pizza and networking. We are lucky to have several members of faculty on our organizing committee, Dr. William Zhao, Dr. Amy Lee, and Dr. Faraz Haq. A number of volunteers, some of which are here today, and others which are at the remote location at SFU. We have 2 more seminars this year upcoming in November and December. Please subscribe to our mailing list if you want to be notified of those. Oh. Yeah, we stopped the screen sharing because the apologies for that. Now we're sharing the slides on Zoom as well. All right, so we'll have a pizza and social networking at the end. This is our VanBug organizing committee, as I mentioned. And we'll have the 2 following talks upcoming in November and December. You can find these both on our website or if you subscribe to our calendar. If anyone would like to do that now, there is a QR code here and as well as the link. We are always looking for suggestions of who you might want to come see speak here. So if you'd like to suggest a speaker for our next season, please use the form on our website to do so. We are fortunate to be sponsored by several organizations, the Graduate Studies in Bioinformatics program at UBC and SFU, Genome British Columbia, SFU Big Data Hub, and the MSL laboratories where we are right now at UBC, Langara College, and Stem Cell Technologies. VanBug is also affiliated with our sister bugs in Montreal and Toronto, as well as the International Society for Computational Biology. If there's anyone participating on the Zoom audience, please ask your questions in the chat and one of the VanBug volunteers will voice it for you. Today, our introductory student talk will be Courtney Hoskinson, who is a PhD student in the Turvey Lab. Her title is Omics in Early Life Gut, Delayed Gut Microbiota Maturation as a Hallmark of Pediatric Allergic Disease. And our featured doc today is Dr. Khanh Dao Duc, who is an assistant professor in mathematics at UBC, as well as an associate member of both the computer science and zoology departments. His talk is Bioinformatic Pipelines, Algorithms, and Nonlinear Metrics for Quantifying Heterogeneity in Large Biological Datasets. Without further ado, Courtney Hoskinson.
Okay. Hi, everyone. Thanks for joining today. I'll be presenting on a recent study led by Dr. Stuart Turvey and Sharice Peterson out of our lab at BC Children's Hospital. This was recently published in Nature Communications in August of this year, and we don't really present at UBC often, so I'm happy to be here today. Explain. Thanks.
Yes, sure.
Okay. All right, thanks for your patience. Now, to give a little background, our lab at BC Children's focuses upon identifying early biomarkers to predict, prevent, and understand allergy. So this is an important field of study as allergic diseases now affect about 20% of the population worldwide. And connecting this to the gut microbiome itself, the microbes living within the human gastrointestinal tract have been linked to the development of allergies by our lab and others, but there's still some ambiguity surrounding how these microbes are affecting the immune system and subsequent allergy. So specifically within this study that I'm presenting today, we looked at multiple allergic outcomes. These included atopic dermatitis, asthma, allergic rhinitis, and food allergy. And so these may seem like very distinct clinical diagnoses, but we decided to look at all of them together due to the fact that they potentially have common immune mechanisms. And these are typified by characteristics like type 2 inflammatory responses and elevated IgE. And moreover, they commonly progress or manifest in a similar manner within people who are showing allergic symptoms. So targeting them all together, we thought could provide a better understanding of how they are both unique and similar to lead to improved preventive strategies. And we were able to ask this question using the child cohort. This is a longitudinal birth cohort in Canada that has followed over 3,000 children and families over the past few years over the course of the children's growth and development from birth. The participants are now undergoing their 12 and 13-year-old visits, so it's been going for a while. But the primary allergic outcomes within this study are allergic disorders as diagnosed by clinicians at 5 years of age in the children. So we also utilize evaluations collected at 1 and 3 years to aid in defining our healthy controls, as well as incorporated baseline demographics to identify important early life factors in the development of allergic disease. Before diving into our microbiome results themselves, we first compared the demographics and early life exposures of 592 participants with allergic diseases, specifically the ones I outlined prior to this. And we compared them to 523 control participants with no allergic symptoms across all the times that they were seen and evaluated at their child visits. So this stringent definition of our healthy controls was a key part of our study because it really increased the confidence that we had in our results by eliminating the confounding signals of the participants with transient allergic diseases. And when comparing the allergy groups to our healthy control groups, we found specific factors that were consistently associated with an increased risk of allergy. So this heat map depicts the odds ratio of developing allergic diseases indicated by shading, and the statistical significance is denoted by stars for an adjusted p-value of less than 0.05. And so we see a particularly impactful effect of antibiotics being biologically male and being non-white. Next, moving on to our microbiome results themselves, we analyzed a few different data sets. These included shotgun metagenomic sequencing and metabolomics data collected from 3-month and 1-year participants' stool samples. So we looked at these data sets to really investigate the microbiome features that might be impacting the development of the immune system and later allergic symptoms. So these data sets allow us to analyze the microbes within the early life gut, which is encompassed within the question of who's there, as well as their functional potential and associated metabolites encompassed within the question of what are they doing. And to start, we first looked at the microbes present within the abundance of microbiota within the infant gut. So before we get to results, it's important to note that changes in the early life gut microbiome occur together with immune development, and microbial components within the gut can affect immune programming. So this is really exemplified in the classical consideration of short-chain fatty acids inducing the differentiation of Treg cells, indole lactic acid preventing the expansion of Th2 and Th17 cells, and so on. So this appropriate progression and fluctuation of bacterial populations is therefore essential to proper immune development. Given this importance of microbial population progressing in immune regulation, we first explored microbiota maturation in the infant gut. Specifically, we derived a microbiome predicted age from species level relative abundances of the microbes. We did this using a nested 5-fold cross-validated random forest regressor. And through the creation of this microbiota predicted age, we found that the structure of the microbial species within the early life gut was so linked to infant development that it could predict the participant age. So this allowed us to compare development across our groups and really identify any irregular patterns that may have been developing in allergic participants. So the central finding really within this paper is that using this predicted age, we were able to discover that participants with allergic diagnoses at 5 years had a consistently lower predicted age at 1 year, despite having nearly identical chronological ages at sample collection. So within this graph, the x-axis is the microbiota predicted age, and the y-axis is the chronological age, with the lines indicating the means of each group. So you can see that the chronological age is nearly identical for the groups, the healthy controls or the allergic participants, while the predicted age, the allergic participants had a delayed maturation metric. So, and importantly, this remained consistent across all the individual diagnoses as well. We next looked at the functional potential of the microbes present, so all their sequence genes annotated by functionally databases. So it just provided insight into what these microbes were capable of, the metabolites of producing, their function, their activity. And that was our next question. So using the Meta-Psych database, we identified pathways that were significantly associated with one predicted age, as well as the outcome of having 2 or more of these clinically distinct allergic diagnoses. And this resulted in this list of 11 pathways meeting our significance thresholds. And this was an important finding because we observed 2 distinct trends within this analysis. The first of these trends was that there was an increase in the potential for inducing an increased oxidative state and mucosal breakdown within participants who would later be diagnosed with allergic diseases. The second trend that we observed was a decreased potential for secondary fermentation, short-chain fatty acid production, and mucosal strengthening within these same infants. Finally, we analyzed 212 metabolites within the infant gut to gain additional understanding into really the broader biological impact that the microbes might be having and to identify targetable biomarkers of allergy risk. An important aspect of this analysis was a simple Spearman correlation between the pathways identified in the analysis I outlined before in combination with important microbiota in our predicted age model. So this is a heat map with red shading indicating a high positive correlation between the variables, while the blue indicated a high negative correlation between the variables. And the primary findings from this investigation included identifying a significant association between pathways associated with an increased risk of allergy and a low microbiota predicted age, with 3 metabolites within the trace amine family. Now, these metabolites are fairly unexplored, especially within the realm of allergy, but they have been seen to influence Th2 responses, increase inflammation within the gut, and together this might potentially exacerbate a more allergic-prone immune system, thereby negatively impacting health outcomes later on. Finally, as a closing analysis, we use structural equation modeling to see whether the associations we identified with our analyses could rise to the question of causality. This is a multivariate analysis using It analyzes structural relationships between measured variables and latent constructs by combining factor analysis and multiple regression analysis. So despite there being no significant direct effect between this predicted age and allergic disease, we did in fact find that the important pathways and metabolites that were different between allergic or non-allergic participants could mediate the relationship between this delayed maturation and allergic disease. And finally, in close, overall, the pathways and metabolites involved in increased oxidation, mucosal breakdown, and Th2 skewing showed trends of increasing risk of allergy, and this stemmed from a lower microbiota maturation metric, while pathways and metabolites involved in fermentation, tolerance induction, and mucosal strengthening showed trends of protecting against allergy via an increased gut microbiota age. And shout out to all the Turvy Lab members, all of our support and funding, and I thank you all for your time.
Now?
Thanks. Just to be clear, your predictor is based upon compositional species of fungus, right?
Yes.
So can you comment on how generalizable you think that your predictive model would be in a different environment? You know, if you're going to take kids from a different country with a diet, and I'd assume it'd be different, but how well do you think you could generalize not necessarily the species abundance, but the actual metabolic outputs or any other kind of facet of what the actual microbiome type?
Yeah, that's a fantastic question. Our metric is definitely in-house derived from the child cohort data set. So the metric itself couldn't be translated directly, but we are working with one lab that's kind of utilizing this idea of colonization over time within a mouse model. And they're looking at the metabolites associated, the different microbes associated with it, and allergy outcomes. So that might provide a bit more insight into the generalizability and the biological impacts of those different factors. Yeah.
Just wondering, do you do any sort of comparisons as to the like a solid food, like introduction of solid food and how it might affect this predicted age onset? Because to me, the data looks so strongly bimodal that my immediate thought is that is this even like meaningful, or is it just an artifact of when parents decide to introduce solid foods?
Yeah, I think that's a great point, especially when you're considering things like peanut allergy, and the introduction of peanuts within the early life can really determine whether you're allergic to peanuts later on. And that general trend of different diets during early life can shape the gut microbiome. I think one of the ways that we control for that is that within one of our sub-analyses, it's in our supplement, but we incorporated the breastfeeding status of the infants and whether they had been given solid food at the time of the visit, and controlled for that when considering the differences between that microbiota age metric. And we did still find significance, but that's a great question. It's not always easy to control for those different variables, but definitely an interesting thing to look forward in the future. But thanks for your question.
Any other questions from the audience? Thank you very much. Perfect.
The micro, maybe? Yeah, okay.
So you want to try to put it on this?
Say it again.
I don't think we have much choice.
If people can hear me, should be fine.
Yep.
Can people hear me? Okay. Yeah. Will I start?
Yes,
Yeah.
please.
Okay,
Yeah.
great. Okay, so first I want to really thank the organizers for putting this together, for the invitation. I'm very excited to be here and to talk to this specific audience. I understand that there are a lot of graduate students in bioinformatics. And basically, this is, since I moved to UBC, this is what you can see in the title is what I've been trying to do, is really to try to develop these pipelines, algorithms, and use different metrics. And we apply that to a lot of different data sets. So when I was given the opportunity to give this talk, I was not sure actually what I would talk about. But then I decided to really focus on what you can see on the background. This is a 3D structure of the ribosome. Okay, so it's a very large complex that is imaged here by another technique called cryo-electron microscopy. And so basically, by focusing on that aspect of my research, I actually want to cover these different methods and pipelines that we put together. I hope that they can really help to solve interesting biological questions. And what I also hope is that in the second part, I can tell you a bit more about the cool mathematics that's under the hood. Okay, so I'll start with a very kind of general introduction of the biological context and what I want to talk about. So these ribosomes. So these ribosomes, they are basically these molecular machines that are mediating translation. That is one of the two parts of the central dogma of biology that says that the genetic information can be expressed into proteins with some intermediate messenger RNA. And this is a dual process. First, there's transcription. So you're transcribing your sequence of nucleotide into a sequence of triplets of codons. And then from these 61 different types of codons, you get a sequence of peptides. This is a protein. And you have 20 amino acids. So this is what this process is called translation. Okay, and if you want to zoom a bit, go to what we call the mesoscopic scale. You get this picture, this cartoonish picture that you see at the bottom, where basically this, in red, you see these ribosomes that get assembled at the start codon, right? So this is a phase called the initiation. And then they would move along, right? So they're basically along this way, there's always for a given codon a specific transfer RNA that will bind. And once it's bound, then they can basically translocate and move from codon to codon until they reach the stop codon. And what you can see also in this picture is that along the way, they assemble this chain of amino acids so that at the end, you get a fully formed protein. Okay? So this is a very, very fundamental and basic process in biology. But when you look at the details across different systems and all the regulation that happens, this is where things become very, very messy and incredibly complex. So let's maybe zoom a bit more. So now I want to go from this cartoonish picture with these red potatoes, right, that are separated into these two different parts, to what you see in real life. And this is actually something that that's really the results of decades of effort that actually culminated in the Nobel Prize awarded in 2009 for the determination of the ribosome structures. And you can see also on the bottom all the progress that was made using this technique called cryo-electron microscopy, where first you got these kind of very blobs, and at the beginning of the development of the technology, actually people were joking about cryo-EM as just a kind of globology field. But you know, with the advance of the techniques, computational methods, you now can get access to visualization of these structures in 3D at a really, really high resolution. So these two subunits that I mentioned in my previous slide, you can now visualize them in yellow and blue. And in green, you see this transfer RNA that is basically carrying the peptide that is part of the protein that the ribosome is translating. Okay, and getting this structure was really concomitant with the development of the technology of cryo-EM itself, and led to another Nobel Prize in 2017, because this has many applications. Now in the field of structural biology, cryo-EM is basically starting to really take over, and from there we can really now visualize and study all kinds of proteins of interest at high resolution, and this is really fundamental for biology. Okay, I want to also, because this is a kind of general talk, I want to tell you a bit more about the cryo-EM pipeline for those who are not aware of it. This is an image actually that was produced by my master's student, Elena, who's actually a graduate student in the bioinformatics program. And I also want to mention it because there's a lot of interesting and really fascinating bioinformatics and algorithms behind. The reason why now we can really obtain these very high resolution structure is really that people were really smart in different, phase of all these pipelines to really make sure that starting from basically very noisy 2D images that are just projections of your molecule along a grid, you can just pick all these particles, try to deconvolve with all the microscope effects, try to classify, get all these different particles, then try to infer the pose, the angle, to try to reconstruct a 3D model, and later on try to really fit an atomic model within it. Okay, so this is really a lot of effort, and structural biologists spend months sometimes to really get a structure that is as detailed as possible. So you want the model that you build to really fit as much as possible your density map, and you want your density map to be as refined as possible, so you can really identify all the different aspects, all the different atoms, the backbones, the different chains. So you can understand, for example, in the context of the ribosome, you can really locate all the different parts, maybe locate a polypeptide, maybe locate a drug, etc., etc. Okay, so if you think about that along this way, for people who are interested in method development, there are plenty of interesting problems, and so I'll talk about this at the end of my talk. But to get back to the ribosome, I think also a reason why it's so interesting to study it in the context of cryo-EM is because it's actually really not enough to get just one single structure of a ribosome. If you think about all the different steps, for example, that are associated with translation that I showed you at the beginning, there are a lot of what we call conformational changes, right? So these subunits, they move together. The tRNAs, they move from different sites. This is basically what I show you on the right. And it's only a very partial vision of what of the different conformations that a ribosome can adopt, right? This is only focusing on elongation, but think about all the other parts. Think about when a complex can bind to mediate post-translational folding, translation arrest, etc. So basically, people are still producing a lot of structures and trying to understand just all the little details of the ribosome machinery. Okay, there's also something that is interesting is to think about now a comparison of the heterogeneity of the ribosome now across species, right? So the ribosome is a universal machine as a part of the central dogma of biology, and so it's you can take a look at a bacterial ribosome or eukaryotic ribosome, and even within these domains of life, you would find a lot of interesting differences. So if you want a big picture, you can take a look at these two structures of the ribosome, one in E. coli and one of Homo sapiens. You can already see some differences if you look at the RNA at the secondary structure level. And these differences basically also translate in 3D, right? So these ribosomes, they don't have the same mass, they differ in their composition, and these differences have really some big impact functionally. So what you can see ultimately on the right is what we call the ribosome exit tunnel. This is basically where your nascent polypeptide will go through during the translation process. And basically these modifications of the different components, the ribosomal proteins, let's say UL4, UL22, UL39, they really shape this ribosome exit tunnel. And this has some impact on the functional level, right? So now we are basically at the point where we're saying, okay, we have this amazing technology, we can get plenty of structures. These structures are underlying different steps, different functions, etc. But what if I want to compare them, right? What if I want to compare, analyze the differences between ribosomes from, let's say, between E. coli or Homo sapiens, or along all these different conformational pathways? Okay, and well, if you were bioinformaticians and you had structures, well, you could say, okay, we can do some alignment, do some pairwise comparison, but in 3D, well, you have to think about other tools, right? Okay, another thing that I just want to mention is just to give you an overview of just the available structures and why this problem has become quite timely. I would say that since Cryo-EM came to what we call the resolution revolution, there has been a surge of structures, right, that came for the past 10 years. So this surge of structures came with a lot of different species and a lot of different conformations. So if you look at the PDB, the protein database, where these structures are available, this is a graph where you can see how basically there are more and more structures, and these structures are also imaged by Cryo-EM. Right, so when I basically started getting interested in translation, that was like 10 years ago, I had no idea what a ribosome looked like. But then, working with sequencing data, I got more and more interested about this interplay between the structure and the polypeptide for translation dynamics. And I realized, oh, actually, I have all this data that I can play with. Okay, so this is something I started really thinking about at the end of my postdoc, and it really started from the following: just simply looking at the PDB and looking at different databases and try to see what we can do if we play with all these structures. Right? So what if we want to compare? For example, I'm taking a ribosome, I'm taking two similar homologous chains, right? So I'm taking one protein, another protein, and I want to say, okay, let's see how they look like. Well, it turns out that if you want to do this, I mean, if you wanted to do this maybe, I would say 5-6 years ago, this process was incredibly tedious. The reason why is because basically if you go to the PDB, the protein data bank, you look at structures available and you look at the different parts of it. The way they're labeled is basically coming with some letters that are just up to the people who deposit the structure, right? So if I'm asking, for example, this protein that I mentioned, UL4, if I want to compare that to another structure, well, I can take a look really at the PDB file. It will be named UL4M somewhere, 50S ribosomal protein L4 somewhere else. Here you will just have a PFAM code. So that's incredibly tedious. So at the end, my postdoc, actually, I spent a lot of time just doing this manually over 20 structures. Okay. But I was just thinking, oh, it would be great to just have students who could try to not do this again, because that would be stupid, but really try to automate that, right? So we can save time along the way. Right, so the first thing I want to tell you about is this first project that really came out after I started my position here at UBC, which is basically trying to build these tools and some workflow to facilitate the comparative analysis of ribosome structures, right? And I really think that this is something that is also, in a way, it's not only an obsession by me, but really something that the field might be interested in. And there was some effort actually that were previously made. I'm mentioning here this nomenclature that we are actually using. And all the people that you see who are co-authors there, they are basically big name in the ribosome field, right? So they just realized, okay, we're building all these structures, but we really want to have a way to communicate with each other, right? But the thing is that even after this was came out, it was still, you know, from a practical standpoint, people would still be depositing these structures using some arbitrary procedure. So our goal here is really try to think about how we can be a bit more efficient. Okay, so what I want to talk to you about today really as really the first part of the research that we've been doing is this database that we call Ribo-XYZ that was published actually in Nucleic Acid Research last year. And it's a comprehensive database of ribosome structure where basically we take all the structures available from the protein data bank. We combine that also with PFAM, that is also a large database of proteins that contains a lot of sequence. And we try to basically come with a new framework for trying to annotate the ribosomes and also provide some visualization tools and comparative tools. Okay, so this is the web application. So what you can see in the bottom is the front end that we developed. Okay, that contains all these visualization tools with the web server. So it's on the top right. And at the bottom, this is the backend when you can have all the data and all the tools. Everything is articulated with an API, which means that you can really also access to these database programmatically via some online queries. And basically, that's basically the general architecture of the database. Okay, some applications that you can actually use if you go to the web application is, for example, you can take different ribosomal components and superimpose them. So really trying to compare specific parts of it. Or you can also, for example, take, let's say, a ribosome that is bound with an antibiotic and try to think about if I'm taking another species where this ligand would bind. So the two figures that we can see here, they are basically reproducing also what we saw from recent papers. So we can obtain the same quality of figure, but we generate that very, very quickly. Actually, there's if you go to the web application, we recorded some videos where you can see that you can do that in 2 minutes. Okay, so I'll tell you a bit more about how the database is structured, some of the tools and ideas that we have. And I think also what I want to point out is that I'm talking about the ribosome, but I think this is a kind of general issue in biology. I think people, when they are looking at some very specific complex proteins, etc., and structural biologists in general, when you produce a structure, I think at some point, if you try to zoom out and try to understand maybe these differences, you would encounter the same kind of issues, right? So I can tell you, this is just a tweet that was posted after we published the paper, where they said, okay, maybe if something similar could exist for the spliceosome, that would be great, right? So think about the RNA polymerase, spliceosome, CRISPR complex, you name it. I think this is a valid and you know, interesting approach. Okay, so I just want to give you just a few more details on what we do, because there's also some bioinformatics behind, and how basically this process is really, how can we automate that, right? I mentioned before that, okay, in principle, you can do this manually. We take a look at the PDB, look at everything, and do that. But how do you automate that if you have thousands of structures, right? So this is what the pipeline that my student Artem came up with. He basically is encoding all the sequence into FASTA files, and then he runs some hidden Markov models, right? These hidden Markov models will generate some classes. And then if we have some reference to begin with, we can really identify all the elements. And then you can just basically go backwards, look at your structure again, reannotate it, and really use the the universal nomenclature that we plan to basically implement and apply for all the structures. Another important aspect that I should mention is once you annotate that and once you got all these chains correctly assigned, another thing that we can do is go in closer details, so not looking at the chains, but more specific parts of them, and try to put some landmarks. So there are, for example, for people who are interested in the ribosome, there are very key parts. For example, what you can see in green, we call it the polypeptide transferase center. It's called the PTC. And this is basically where the tRNA would bind and really form a bond with the nascent polypeptide chain. Right? This is basically where elongation happens. There are other parts, right, where the chain will exit. There are different stalks that can bind with different complexes. And so what we can also do with our approach is really now produce these landmarks for some further comparative analysis. Okay, and let me talk now about some very concrete application that actually came out from my previous research on the dynamics of translation. So I want to talk about this compartment of the ribosome that is called the exit tunnel. So I mentioned before that when you look at the ribosome structures, you have the tRNA that binds and connect with your nascent polypeptide. But if you want this process to happen, basically you want your peptide to basically go through your ribosome and just exit, to form. And then when elongation happens, you get, a full protein. So along this way, at early stage of translation, your nascent polypeptide basically go through this cavity that we call the exit tunnel, that is 100 angstroms long, and that can contain approximately 40 to 50 amino acids. And it's a very narrow region, and it also means that there can be possibly interactions between the polypeptide chain and the ribosome itself. Okay, so we call it the ribosome exit tunnel, and if you're looking on the right, what you can see on the right is what we call the Sec-A pathway. So it's an arrest translation pathway where basically by translating a specific sequence, you can just stall the ribosome. And then this will basically release your mRNA chain, allowing for some subsequent translation run downstream. And so there's a whole pathway that is associated with translational arrest. So basically, it's very important to understand this interplay between the ribosome and the nascent polypeptide chain for translational arrest, but also co-translational folding. There can be some structure that is acquired by the polypeptide within the tunnel, etc. So for me, it was really like a really key concrete application of having all these structures available and trying to say something about these differences. Right? So the point of also getting these landmarks is that if you want to extract this tunnel, now you have a starting point. Right? If I'm giving you any structure available and I'm asking you to find and locate the exit tunnel, I will at least get one piece of information. I will locate the PTC at the start of the tunnel, right? Something that you can do manually, but if you have thousands of structures, that's not possible. And once you have this polypeptide PTC, you can again use some algorithms, run some bioinformatics. So you can basically try to build, we call this tunnel search algorithm. You try to build a set of spheres that will basically try to get to the exit. And once you get to the exit, you really get a fully formed tunnel, right? And then you can get properties. You can analyze what this looks like. for some downstream analysis. Okay, so this is something that you obtain if you do this for the set of structures that is detailed on the right. You can look along, if you move along the tunnel, you get this set of spheres, and you can think about how the radius is changing, right? And from what you see in this plot is that you can see some interesting variations. There are peaks and valleys, seems to open, and this now gives you a kind of phenotype. You get all these species, now you get all of these radial plots, and now you're getting a bit closer to understanding differences between ribosome exit tunnels, right? Another thing I think that if you want to understand what we're doing here, we're starting from the ribosome, we're starting from a 3D structure. Then we really focused on this exit tunnel. And then we extracted this set of radial variable, basically this radial plot. And so we're really reducing all this geometry now into just one plot. Okay. And now what we can do is now you can try to compare this plot together. You have really like a tool to do some comparison. You have a metric that you can apply, and then you can try to understand and measure and quantify differences. Okay, and so if you do that, well, you apply some hierarchical clustering methods, for example, by looking at, let's say, there's some average distance between these plots, and you get the tree that is shown on the left. And at the time, it was really remarkable to me that the kind of clustering that you get is actually very close to the phylogenetic tree that you would get from sequence alignment. Right? So it's obtained, what you see on the right is obtained by just doing some phylogenetic inference using sequences. And what you see on the left is just using this geometric distance between the tunnel. So that suggests also that there's really some strong coevolution and selection along this way to really try to, that really basically separates these different species and different domains of life. Okay, so we studied that into the details, and I don't have the time to really go into it, but I think this was really something that excited me at the time, was really to be able to connect this geometric trait with what you do when you think about molecular evolution. Okay, so this is a result that is unpublished. This is what we get now when we add some more species using our database. And you can see also some new patterns that emerge, right? So before, I had a very clear separations between eukaryotes and bacteria. And now we found these two more recently produced structures that now cluster with bacteria. And this is also some ongoing analysis, but we analyzed the tunnel, and it turns out that if you look at the landmarks that really characterize these two tunnels, you really have something that is closer to a bacterial tunnel than what we see for the rest. So this is still going on, but what the goal is really now that we have this tool to automate the extraction of the exit tunnel and this idea of comparing these shapes, now we can even produce some new results. Okay, and why not also thinking about something that I would say even more refined? How about building a real evolutionary model, right? How about trying to think about how the shape could really evolve in time? This is something that people do if they are looking at phenotypic traits. So like, let's say this tooth shape, this is coming from a paper where you basically look at different tooths from different species, as well as this landmark, and then you can start to build an evolutionary model, find some common ancestors, and try to maybe visualize some evolutionary events, right? So this will definitely require some more tools, extracting the shape at a higher resolution, not only looking at the radial variation, but also thinking about defining this landmark, and at the end, choosing a right and appropriate what we call morphometrics in the field of evolutionary biology. And in the same way, you can think about all these conformations of the ribosome. So I'd like to get back maybe to what I mentioned before, all of different intermediate structures, how can we organize them? And this is also a very recent paper that came out that really tried to organize all the intermediate structure according to their molecular weight and try to build some hierarchy along that can say a bit more about what's the pathways for assembling the ribosome. I remember I spoke a few years ago with a professor who's really a specialist on ribosome assembly, and he tried to describe me everything just on the whiteboard, showing how an arm could fold, generate a scaffold, move everything. And at the time, I think even a few years ago, when people tried to think about all these assemblies, it was basically all getting these structures and just trying to figure out things in manually. But now, if you get all these structures, I think there's a need for an opportunity for some more automation. And again, I think the thing that you want to think about here is what metric you would use to really compare these structures together. So again, we're getting, I think, to this original question that is for me just a mathematical question. If I'm building a set of objects, how can I really model and compare them, right? And even without thinking about these very specific applications, this is something that has very basic applications in the field of, let's say, in the context of interpolation or alignment, right? You get two different objects. How do you align them? How do you generate some morphing between both? This is basically all that depends on the choice of the metric. So this is getting to the second part of my talk, and I want to now give you, I hope that there was enough specification to do some math now. Okay? So I think for the past few years, I've been thinking about this question, and basically, if you want to really do a standard comparison, you can choose, let's say, a Euclidean distance. This gives you some results. We call this the linear distance, but maybe there's a way to be a bit more creative. And the advantage, I think, of knowing a bit about mathematics is that you can get access to all of these more complicated tools that are nonlinear. And one specific tool that I want to talk about now for the second part of my talk. So what I want to describe is a problem that came actually 300 years ago and have some very, very recent development with some more the advance of algorithms and computational methods. So this is a problem that we call the problem of optimal transport. And it basically tries to build the distance between distributions. So the kind of example that people come with in general is the following. Let's say this is a map of Paris. And in red, you have different bakeries, and in blue, you have different restaurants. Okay, and these bakeries are making bread, and they can basically choose to deliver this bread at different locations, right? So if you have a bigger red point, it means that it produces more bread than another one that would be a bit smaller. Okay, so the question is, how do you, and assume also that these restaurants have different demand, right? Maybe a restaurant has more capacity, so it would need basically more bread. So the question is, how can you distribute the bread in an optimal way that could minimize the cost of, let's say, moving in Paris? You want to deliver that, and you want to do it in an efficient way. So this is something that people, actually Russians, mathematicians, think a lot about this in the context of operational theory. But I think this has many and many applications. And if you look at our recent publications, we not only use that for cryo-EM, but for electronic health records. We're thinking about using that for cell images, etc. But if you try to formalize that, basically you come into looking at these two distributions, right? And in between, you can define some costs, right? That could tell you, okay, how much it costs to move from one specific bakery to another restaurant, right? And so the way you can distribute that, define different strategies, and you can try to minimize that. And the result is this equation here, this optimization problem that defines this distance. Once you try to minimize that, when you try to find the minimal cost gives you a distance between these two distributions. Okay, so how do we apply that to cryo-EM? Well, the reason why I want to apply that to cryo-EM is the following, is that if you want to build, now that you have a distance, if you want to build what we call geodesics, So if you want to connect 2 points with the shortest path according to the distance, now you can really see the intermediate structures coming to, along this path. And the intuition here that you should have is that if you produce a geodesic in this space associated with the transport-based distance, you get things that move really, the mass gets transporting along the way, right? This is a transport-based metric. Now, if you use a classical distance, what you would see is the following. You want to transport this mass into this mass here, but what you would do along the way is just teleport one part to the other part. This is just teleportation of mass that you can see along this geodesic here. So if you want now to get back to the idea of having two structures together and try to understand how you can maybe morph one into the other or analyze the difference, my claim is that it makes more sense to think about this difference in terms of transportation of mass, like physical mass. So it also makes more sense to use this metric to calculate, let's say, interpolants or to compare different densities, right? So this is basically what we implemented, and the result is the following. If you look at these two structures that has two conformations, one is closed and the other is open, you can see that if you proceed with linear interpolation, you see that the results in the middle is just blending the two objects together. But with our method, you can really see the arms just moving together. So if you want, if a practitioner wants to visualize how, I don't know, how a chain is closing, opening, or if there's some other conformational motions, this is, I would say, more physically suitable to. Okay, so another application could be for alignment. Let's say you're given two different maps, right, here, that have some different conformations, and you want to align them. So again, you can use the same metric, solve an optimization problem. And we did that a bit more recently with my student Ariane. And we realized that if we use the standard method that is implemented in Chimera, you would sometimes get stuck into some local minimum. So you can see here that basically when you try to align the map in red and in blue, what you get from the Euclidean distance is, okay, first you put things in the same axis, right, the main axis. But then because you've already done a good job at it, it's hard to move things, right? So you get stuck in this local minimum where basically everything still needs to be rotated a bit. Sorry, is it no? Okay. And what we obtain is basically using this Wasserstein distance, we're able to move things a bit better and get better alignment. Okay. But there's some assumption here, and I think that's a major problem if you think about these intermediate structures or alignment in general, is that there's assumption here that you basically want to transport all the mass of one map into the other map, right? And this basically arises from the formulation that just comes from these equations. It's basically there's an assumption here that everything you produce, you will transport it to your whole target distribution, right? So how does it work? For example, if I have just a subunit of the ribosome or a partial structure and I want to align it. Well, this would not work for what we do here, but this is our most recent approach, is now thinking to reformulate and relax this optimization problem and think about using another type of distance. Okay, and I don't want to go into the mathematical details, but the underlying distance is called the Gromov-Wasserstein distance. And the idea is now not only to directly map things into the other, but first think about some internal distance between your two objects, right? So let's say I have these two landmarks. I know that they're separated by this distance. And if I'm looking at the other structure, it would be another distance. And now I have this whole internal structure, and now I'm trying to map things together. So that's the very general idea. And again, I don't have the time to detail the map, but we came with a formulation that actually came from some other work. Try to apply that in the context of cryo-EM. And you can see the results on the bottom left, where now you have this complex here. We took this as an example, this dimer that have these two chains. And if you want to map one into the other and try to apply a standard method, you would obtain something very bad, something like this, right? Because your algorithm is just basically trying to match things, ignoring the fact that there's only some partial alignment that you should obtain. And you come to obtain something like that. But now with our method, what we do is that we produce this mapping from the formulation that we produce, and then we get what we call the partial alignment. Okay? So to me, that's really the next step that can make this type of methods actually more broad and more interesting for practitioners. Okay? So let me conclude also with some concrete application, and that's coming from this most recent work, and this is for atomic model building. So I don't know if you remember, but when I started my talk, I discussed about this whole pipeline of cryo-EM, right? And so at the end, there's something that is very important, that is the atomic model building. Once you get the density map, how do you try to fit a structure within it? And usually this is a very big bottleneck in all the cryo-EM procedure, right? People are getting this map and then they try to position all the atoms together. And so there's some really recent effort, I think, to try to automate that, right? And this is an example of a paper that came up with where they use deep learning to do that. And so what they do is they use the neural network to generate this intermediate, they call it a main chain probability map, and then they fit some candidates to try to see how this will work, right? So something interesting also here is that the way the prediction is generated here is using AlphaFold. So I think you can see also the use of now AlphaFold and all these AI tools really coming into fruition, right? And I don't think this approach would have been possible, let's say, 5 years ago. But along the way, I think why this paper is interesting for me is that basically there's something here that happens that is very similar to what I just described before. You're given a general map, you have all these predicted chains, and then you try to put them together. So that's something that we try to replicate now using our own method and not some standard linear method. And actually, we can really do this. We just generate some AlphaFold prediction that generate a partial map. And now if you're given an input density map, you apply our whole framework. You can basically try to fit these two elements partially and build an atomic, get as an output an atomic model. And I don't have enough time, but if we did some benchmarking, and what we do here is actually outperforming other methods for what we call rigid body fitting, right? So you don't get all the details. There still needs to be some tweaks because your prediction to begin with could be a bit inaccurate, but I think this is really a first step to the whole pipeline. Okay, so I just stop here, and I just want to acknowledge the different sources of fundings, but also all my collaborators and also my lab at UBC, especially the work of these two students, Artem, who's in mathematics, and Aryan, who's a PhD student in computer science. I had the chance to have a very diverse and talented lab with people in computer science, chemical and biological engineering, mathematics. And it's been amazing to be at UBC and to be able to collaborate with all the people that are listed here in different departments, and also these collaborators across the world. Okay, so I'll stop here. I really want to thank again the organizers for the invitation. Thank you for your attention, and I'm happy to take questions.
Thank you. That's interesting. I had this question actually first during the database representation of the structures, and also for your modeling of the structure. So if I'm understanding correctly, a lot of it is, for example, for the ribosomal complex, you have consistent subunits, but you're looking at their evolution through their function of the different conformations. How applicable do you think these methods are if you have protein structures with variable subunits that change. I'm thinking specifically something like the mediator complex where you have moving parts throughout its functionality. Like, is this something that you think that you can adapt to where there wouldn't just be the same protein subunits that, you know, you're introducing basically new subunits throughout the process?
Yeah, I think in principle, I don't see any. Okay, I don't see any conceptual issues in doing that, and the reason is that basically, if you're working with if you're working with density map, that's another question, because in density map you cannot really identify. You know, you just basically get the 3D image of intensity. You don't know what's there. If you have an atomic model, you can actually see, okay, this is this protein, this is this chain, I can align them. So even if it's moving space, it's conformational, you still have a way to identify that. So the really, I think the goal, I mean, so there's no conceptual issue. And I think once you do the kind of hard work of trying to, you know, connect the 3D structure, the sequence, and build a kind of command nomenclature language, basically you can do what I did and apply that to any other complex.
Very nice. So, what do you think is the major evolutionary pressure on the dimensional taxonomy? Is it rotational speed? Is it composition of the proteins in general? Do you have any idea?
Yes, so we try to so that's the first. I think what's interesting is that you look at the conservation, you really look at all the components in the sequence. The parts of the core, they are much more conserved than the parts that you. So I think, you know, if you look at this, and it makes sense, right? If you try to have this complex object, it's much easier to treat things that, you know, in the exit at the core. So the core is actually very conserved, but why you get the most interesting variation, but you still get some very specific position where you get interesting locations. So to me, there's this part where we call it the constriction site, where everything gets narrowed, the tunnel basically narrows down. And we found some very strong evolutionary signals that basically explain the separation that you see. So the advantage of this metric is basically you can divide into different regions, and you can see what are the main drivers of the diversity. And so I think there's a specific location of the constriction site where basically I think there's a lot of interaction between the chains and the tunnel that happens. And at the exit also, because you have this binding to different complexes, because you can have some structure that happens, it makes a lot of sense to have some diversity and some maybe some selective pressure for, I don't know, some targeting, you know, different complexes. So that's basically how I can answer your question. But I think that's still ideally, you know, I was saying if I had a model where I could really try to model evolution, extract some time, look at different evolutionary events, that would provide, I think, a much more satisfying answer than what you just asked.
I was still along the line is asking that if you have the model, you have two different conformations, and you're trying to model, you know, the protein structure changing or ribosome subunit changing, and put the new methods the vessel size. So you can try to identify one component and another component, because Cryo-EM is going to have a lot of different densities. And then you're going to try to, you know, get a structure using the,
I can tell you.
So you get a structure using this kind of mapping back to the Cryo-EM's densities, right? I wonder if this, because if you use AlphaFold, for instance, these structures can be predicted at the kind of final stage where, you know, like kind of native stage, not the ensemble heterogeneous of the structure. You think the limit that is limited this by mapping with the cluster style and reduce the kind of density distribution?
So I can tell the article who have tried to use that. So what I'm talking about is what you get when you already have the density map. So I'm working, the density maps have been introduced. There may be some conformational heterogeneity behind from the data, but what the practitioner did was really to find different classes and say, okay, you have that now, do something with it. Now, I think what you're suggesting is, let's go back to in the workflow and think about all these 2D images. There may be some conformational heterogeneity underlying, all the images that we get. Will it make sense to use this? And I can tell you that people have been trying to think about it. I think it's a very hard problem, but I think, yeah, basically you can take a look at the research from Amit Singer and Princeton. He's been trying to bring this sort of, this Earth, we call it the Earth over distance, directly analyzing the 2D images. But that's a bit different from what I've been doing here. I think it's also a bit more challenging, so I'd rather stick to basic.
Other question that you as you was alluding to try to determine the bias side, like for drugs like antibiotics, biotics targeting of the ribosome. Can you elaborate how to do so?
Okay, I think let me elaborate a bit more. I think the application I'm talking about is looking. So let's say you have a ribosome, like a ribosome from a specific species. And then you're thinking about, oh, I have a drug with a bind on it. And then you can, for example, use docking methods, MD, etc., to do that. But you have this big structure, and so you would like to know, okay, where should it bind? So what I'm saying is that if you already know that this ligand will bind in another structure that exists, you can just leverage that to just say, okay, let's take a look at the corresponding site in this structure and try to see if, it just basically it's a way to facilitate that, right? So that there's room for much more methods after that, but I'm not an expert in that. But I'm sure you are. So maybe
to have some kind of detecting binding pockets, because the novel binding pockets, the current pockets just target the where the RNA binding.
Okay. So in this case, I can tell you, like for antibody receptor, I think I can think about this in the context of antibodies. All these binding pockets, I think if you think about tape analysis, what would be super cool is to develop this method to really try to find similarities in terms of the binding sites, and then apply that. Actually, there's some interesting math that I can that I've been thinking about, but it's a kind of a different problem, but I think there are ways to address that. Yeah. Predicting binding pockets if you have a lot of different structures.
We have a question from the remote audience. You want to ask that?
Hello, can you hear me now?
Yes.
Yeah.
Okay. Hi. Thanks for the presentation. This is Will Shell from SFU. I was just curious, when you were the first part of your talk where you presented the sort of correspondence between the ribosomal structure and the phylogenetic tree based on 16S. Did you notice any discordant branching patterns? And the reason I'm interested in that is just whether that sort of discordant branching pattern would point to maybe sort of, so I'm interested in horizontal gene transfer. I'm wondering if there's any sort of grouping that's at the structure level that we can detect based on, traditional phylogenetic analysis, and therefore use that to sort of detect maybe certain organisms' DNAs are more compatible and express better in certain other sets of organisms. Thanks.
That's a very interesting question. So the correspondence is not perfect, and so what you can see here on the left is what you do, at some degree of clustering, right? So if you get a bit, if I'm detailing the tree that I would obtain here, then I would start seeing some differences, some more noticeable differences. So I haven't thought too much about what you described, and for the following reason is because I think the metric that I chose to do that is still very rudimentary in the sense that I'm looking at just this radial variation, right? And I'm building a distance that really aggregates, you know, all these variations into just one measure. And so if I wanted to detect some much more subtle signal, I think I would be in trouble, and I would need something that is a bit more elaborate. And so that's why I think what I described here, where people are just selecting fuel and marks, and that's something that we could do for the tunnel. Now that I know some region of interest, like Jörg was asking earlier on, maybe with some more advanced model, right? That would basically transpose the framework that you can see in morphometrics, like for shape analysis, into the ribosome. I think we might be able to find some, you know, like the discrepancies. I think that would be much more interesting to interpret, and maybe, yeah, in the context of horizontal gene transfer, that would be amazing to think about that. Yeah, thanks for your question. It's really a great question.
Great, thank you.
We have time for one last question. If we can one over here first. We can repeat the question.
Okay.
Yeah, I was interested in when we were using optical transport to push the density of, it looks like a 4 through time. I was wondering to what degree like you guys constrain that by thermodynamic like requirements, or not at all. Okay.
Totally, it's just the math. Digital optimization program that is.
Okay.
How do you basically transport the mass in blue to the mass in yellow in the less costly way, where the cost is driven by the ambient distance. So I'm saying that if it's, I'm assuming everything moves at uniform speed. So basically, if I'm far away, it costs, basically, if I'm twice more far from a point to another, it will cost me twice more to get there. So that's basically what you're trying to solve. Now, what you're saying is maybe that if you add some more constraints, like based on the physics, right, and the structure, I think you could actually identify more realistic factors. But that's also an interesting question. If you have, let's say, some observation from MD this year, maybe you can also try to kind of decouple, you know, the thermodynamics effect, which is geometric effects. So that's something to think about. You can also tune your cost function. This is a very flexible framework, so you could possibly maybe tune your cost function to add some more physics.
Cool. Thank you.
Great.
Oh, sorry, I didn't repeat the question. It was
Just a
yeah.
thanks to work out.
Okay. Yeah.
Thank you very much. Everyone's looking. There pizza and drinks across that SL 101. Thank you.